So this session, just to introduce myself, my name is Raksha. I work at the RSB, and one of my responsibilities is to oversee the Plant Health Undergraduate Studentships program. So the Plant Health Undergraduate Studentships program is a summer studentship scheme where students from UK universities can apply to be a part of a plant health project at one of the host institutions. So Tia has very helpfully posted a link in the chat if you're interested in finding out some more information. But overall, the plant health undergraduate studentships are a great way for both supervisors and students to be a part of a summer program investigating one of Defra's plant health priority themes. So during this session, we're going to have some previous students and supervisors who have partaken in the studentships program, who are going to share their experiences and projects, so you can see the value that they had in the studentships. So these are really valuable opportunity for students who are in the middle years of their degrees to see what it's like to be part of a research project, and to gain that valuable research experience, which they might not be able to get otherwise. So usually there are at least 6 projects that are held each year, which is due to the core funding that we receive from Defra, but there are also additional projects that take place. And last year we had an additional 6 projects thanks to funding from the British Society for Plant Pathology, the Association of Applied Biologists, SCI Horticulture Group, and the Colgrave Seabrook Foundation. So there is further information, but if anyone is interested, we run these schemes every year. So if students and supervisors are interested, the programme will open again next year for next summer. And if anyone's interested, in sponsoring as well, we'd be more than happy to. So to have further participation. So we'll pop the register's email address as well for you to get in touch if you're interested or have any questions. So we're going to highlight some of the studentships as part of this session and work that's come out of it. Unfortunately, there won't be time for question and answers in this session, but if you do have any questions for the speakers, please do reach out to them, and I'm sure they'll be more than happy to answer your questions. So the first talk we're going to have is from Rashid. So Eden, if you would be happy to start.
Hi everyone, my name is Rashid. I am a postgraduate currently pursuing my master's at University of Edinburgh in biotech. This short presentation is work I did last summer on the University of Greenwich, funded by the British Society for Plant Pathology. My studentship was investigating the effect of agrivoltaic skewed light spectra on Botrytis cinerea development on strawberry, which is a mouthful, but I will try my best to explain clearly. So what is agrivoltaics and skewed light spectra? Well, agrivoltaics is the practice of using the same land to grow crops and to generate solar energy at the same time, thus making better use of land. In practice, solar panels are designed to track the sun to allow for sufficient energy capture for electricity production, whilst being rotated or tilted periodically to allow sunlight to reach the crops below. Going one step further are semi-transparent tinted solar panels. These panels basically separate or skew the light to where the high energy blue light wavelengths are used for electricity generation, whilst the other wavelengths, including the reddish and orange light, is allowed to pass through to the crops below, hence why these solar panels here are orange. Despite its creative design utilizing light, investigating the effect of semi-transparent photovoltaics on crops remains limited, especially on its effects on growth and susceptibility to pathogens. And this leads to the question, is there an effect? Could skewed light spectrum make plants more susceptible to disease, or could it potentially improve it? Next slide, please. Before diving in, I wanted to briefly introduce the state of literature on the effects of light. It is well documented that blue and red light play central roles in growth, development, and regulation of plants, notably in photosynthesis, photoperiod, and the triggering of defensive pathways. Focusing on these semi-transparent solar panels, recent work has demonstrated that protein content increases in both spinach leaves and basil. Although effective in increasing protein content, skewing the light may have other effects, which can be a trade-off for plant growth. For example, one phenomena I wanted to highlight is called the shade avoidance effect. This is when plants channel their resources into growing very tall to overtop their neighbors to outcompete them, to basically ensure that they can forage adequate light. This response occurs when the phytochromes, which perceive red and far red light, detects the ratio of red to far red light is reduced, triggering the onset of a complex cascade that results in the upregulation of auxin and elongation of the stems. Although effective in survival, the literature suggests that this may stress the plant as it uses lots of resources into growing tall, and thus its allocation to other pathways such as defense is limited and therefore may be more susceptible to pathogens. Increasing the complexity, like plants, fungi also perceive red light through phytochromes. Although the effects of this reddish-orange light have been reported, they are conflicting. Some report that red light induces conidia formation, the asexual spores, whilst others report it inhibits it, and thus is species-specific. I therefore wanted to help elucidate this gap in knowledge by designing an experiment that investigates the effect of different light sources on fungus and to potentially observe any interactions between plants and pathogens. Next slide, please. So the reasoning for the choice of plant and pathogens, the sad reality is that 33% of these horticultural crops are never consumed post-harvest, and Botrytis cinerea, the common grey mold that I'm sure we're all familiar with on strawberries, is the most common post-harvest disease. Strawberries as a plant was chosen as there's been a 142% increase in the production, and there's been interest in LED/red light recipes as they can potentially enhance its sweetness effects and therefore allow to be sold at a further premium price. Next slide, please. So yeah, these are my aims. I set out wide aims for this project, which focused on investigating the growth and development of 5 Botrytis subspecies on 3 strawberry varieties under orange light. I wanted to basically know if there was any effects of this orange light and whether these effects are due to changes in the plant, fungus, or both. I noticed that the plants in the orange greenhouse were taller than the plants growing under no light restriction, which allowed me to hypothesize that basically these plants are shade avoiding. And because of this, I thought that perhaps these plants could have their defenses weakened. I also wondered if this skewed orange light could play a role in the colonization of strawberry, or a role in the response to colonization, or perhaps both as well. Next slide. So this is my experimental setup for monitoring botrytis growth. To attempt to answer my questions, I agree that botrytis variants on PDA plates in growth chambers with the orange film placed on top to view if the orange light had any effect on botrytis growth. To do this, I researched the preferred growing conditions and confirmed the spectral power distribution of the orange film, which peaked around the orange and red wavelengths. Images were then taken in image J, and so I can calculate the mean area of growth. Yeah, so this is my results for BC8, which is one of the subspecies. After 3 days, there was no statistical difference between the control and orange plates, which may suggest that the orange light isn't playing a role in mitigating any botrytis development here. This is also evident as after 1 week, all subspecies under all light treatments grew to the maximum in their petri dishes. Despite this, I also thought that orange light could be playing a role upon the plant pathogen interaction. And to investigate this, I designed fruit and lesion assays. I took new leaves and fruits from the different greenhouses, sterilized them in bleach, and placed them in petri dishes containing damp filter paper and cotton wool to encourage botrytis growth. I then inoculated them with adjusted spore count and took photos every day to track colonization. Mean lesion area was then calculated using image J and then statistically analyzed to see if orange light may have any effect on colonization. Next slide, please. In practice, it was proven quite difficult to measure the interaction effectively. I was a tiny bit hasty into investigating so many variables, which may have skewed some results by not having enough replicates. And if I was to repeat this, I would reduce the amount of variables. And also score the data from yes or no instead of using lesion size, like mean area lesion size. There was no overall statistical significance for most variants under the light conditions when considering lesion area size, but this might be different in the future. However, I wanted to highlight BC9 here, which showed a statistical difference in interaction. Here, under the orange light, it appears that orange light might be playing a role in preventing botrytis colonization upon interaction, and I do think that this would be interesting to pursue further. I wanted to highlight this example in particular as it demonstrates the complexity of biology and how abiota may have different effects when studied through interaction rather than in isolation. Next slide, please. So this would be the next steps, quantifying botrytis development using quantitative PCR, although I didn't get around to doing this last summer. I did design working primers that amplified a gene called BCPG1, which encodes for an effector enzyme called polygalacturinases, and these basically degrade the cell walls by breaking down pectin. I chose this as this is constitutively expressed in botrytis and could therefore quantify development. I also designed the corresponding strawberry primers that amplify a gene called FAPGIP, which encode for a protein that inhibits the corresponding polygalacturase enzymes. And lastly, I wanted to quickly mention that these strawberry genes are only expressed in the presence of the BCPG genes. Next slide, please. Yeah, so in conclusion, there wasn't too many meaningful differences between the light conditions, suggesting that orange light might not be having an overall effect, although I do think it's still of interest to investigate their interaction under orange light. Yeah, next slide, please. Lastly, I would like to thank the University of Greenwich and Dr. Thompson for her guidance and supervision, colleagues Dr. Ferreira, Dr. Wang, Emmanuel and Pa for their support. I would also like to thank the BSPP, RSB, Innovate UK, and the Biotechnology and Biological Sciences Research Council for assisting me. Here is my contact information if anyone wants to reach out for any questions or future collaborations. I'll also put my LinkedIn in the link in the chat, so you can access it that way. And thank you all for listening.
Thanks very much for that great talk, Eden. I think it's a lovely example of showing what the plant health undergraduate studentship schemes can achieve, and certainly have managed to achieve a lot of work in the time. The studentships they run for about 8 to 10 weeks, so that's a large amount of work for a studentship for a student to undertake in that time. So it's amazing to see. And I think we'll all be looking at our strawberries with a lot of appreciation going forwards. But thank you. And as mentioned, please do reach out to Eden if you have any questions on the projects. That was really interesting. So the next talk that we're going to have is by Alice McAllister. Unfortunately, they couldn't be with us today, but have sent in a pre-recorded video because they still wanted to take part and showcase the Plant Health Undergraduate Studentship Scheme. So we will share the recording now.
Hello, my name is Alice, and last summer I worked with Naomi, David and Alec at Canterbury Christchurch University, developing primer sets for virus identification in dahlia plants. The project consisted of 2 main project aims, to develop primers to identify viruses in dahlia and to reduce virus-free plants from infected parent plants. The dahlia are ornamental plants native to Mexico and Central America. They are important economically, and they're often propagated from tubers, which the virus will often infect. So the virus is often carried from the parent plant to the new plant. Common viruses infecting dahlia include dahlia mosaic virus, tomato spotted wilt virus, and tobacco streak virus. And during infection, they produce symptoms such as chlorotic leaf patterns that you can see in this image, but some plants display no symptoms at all. So this project consisted of 3 main methods. To start the project, MySeq data was collated by extracting RNA from dahlia leaves displaying viral symptoms. cDNA was produced and sequenced using Illumina MySeq, which is a non-biased sequencing method which shows all the RNAs from a sample, so that will also include plant RNA and so on. The DNA was then compared to the NCBI database using the BLAST function. And using the MySeq data, primers were designed to the viruses identified and used in PCR with cDNA produced from RNA extracted from infected dahlia leaves. The PCR was ran on an agarose gel, and fragments of expected sizes were cloned into the PCR 2.1 cloner vector and transformed into competent cells. Using blue-white selection, colonies were picked and grown up and then used in a colony PCR, which was then digested with ECHO-1 to double-check the transformations were successful. If that was the case, the PCR was sent for sequencing and the sequence was aligned to the MySeq data. If the data aligned, we were able to validate the primers, which can be used on new plants derived from the virus infected. Parent plants. And to produce the new plants, meristem was removed from parent plants, as viruses don't usually infect the meristem, and that was placed on culture containing growth hormones. And when the new plant grows, they will be tested with the validated primer sets, but this is still ongoing. So these are the viruses that were identified using MySeq. We were able to identify a potyvirus and a fabavirus that we think might be new viruses. And we were able to identify tobacco streak virus, tomato spotted wilt virus, and Dahlia mosaic virus. I've added a couple of diagrams of Dahlia mosaic virus, a potyvirus, and a fabavirus, just to show how different the viral genome is and different structures they have. So here you can see the Dahlia mosaic virus consists of just one particle with double-stranded DNA, whereas the fabavirus consists of two particles with two RNAs. And for us to validate the primers, we needed to align each genomic region to the database. So these are some of the results from the primer design and validation steps. So this is an example of an agarose gel with a band of expected size. The PCR product was cloned into the PCR2 cloning vector, as you can see here. And then the vector was taken up by bacterial cells, and cells which contained the PCR formed white colonies. There you can see the white and blue section. And the white colonies were then grown up overnight in LB media, and they were used in ECHO-1 digest the next day, which cuts the product out of the plasmid. And here on the vector map, you can see the ECHO-1 sites flanking the PCR product. And on the agarose gel, you can see, sorry, my head's blocking some of it. You can see the vector at the top and the PCR product at the bottom. The PCRs which had bands of expected size were sent for sequencing, and this is an example of data that we received. So using Sequentia, we then trimmed the sequence and aligned it to the original MySeq data. And here at the top, this green line, you can see that's the MySeq data. And then here you can see my PCR product in both the forward and reverse directions aligned to the MySeq data. So this is a summary of the primers we were able to validate over the summer. To validate a primer set, we needed a PCR product of expected size, a successful clone, and sequence that aligned to the MySeq data. We designed primers for each genome of the virus and a primer set for the coat protein, as the coat protein is highly expressed. And as you can see, we were able to validate primer sets for each genus and coat protein area of the illavirus, tospovirus, and the novel potyvirus. The ongoing work consists of testing the parent plants and new plants with validated primer sets and continuing to design primers for the remaining viruses identified using MySeq. As a part of this studentship, I was required to produce a detailed presentation for the Colgrave Seabrook Foundation and a poster and short speech for SCI. Overall, I feel that this studentship helped me gain valuable hands-on experience within the sciences and solidified my passion for research. I've now completed my degree in plant science, and I've secured a PhD position at a large research centre, and I feel that this studentship helped me to achieve that. I'd just like to say thank you to the Royal Society of Biology for arranging the studentships, and to the Colgrove Seabrook Foundation and SCI for funding this studentship. I'd also like to thank Alec, Naomi and David from Canterbury Christchurch University for this amazing experience, and thank you all for listening. Thanks.
I know Alice obviously couldn't be here, but that was a really great talk. I think we'll all agree, and it's lovely to see the note about the value that she had in the studentships, and it's great to see that both of the students that have presented so far have gone on to further research as well. So I think that's a really good testament to the studentships programme and where it can go, and the broad range of research that can undergo in the plant health umbrella. So yeah, really great talk so far. So next up we have Kelis Fisher. So Kelis, if you'll be able to start your talk now.
So hello, I'm Kelis Fisher, and I did a RSB studentship last summer. So I worked at NIAB with Nicola Hawkins, where I did a 10-week studentship, and I sequenced different rust samples from all across the UK, where I sequenced brown rust, yellow rust, and barley brown rust. So I created primers that had this that looked for this specific sequence to identify different mutations that were linked with fungicide resistance, so that we could really understand where these strains that had fungicide resistance were popping up all over the UK. So the main skills I really gained from this was creating primers. It was the first time I'd ever used softwares like Genius, so it's really helpful to have Nicola guiding me through that process. I also did lots of DNA sequencing, which has really helped me with the projects that I've been working on recently. I also had the opportunity to do some field work, so it's really nice to be out in nature. And I also was able to present my findings at the end. And again, this was a really helpful experience, as I was able to work in industry, both in labs and in the office. So it really was very helpful, and it made me feel a lot more confident. So since then, I graduated from the Uni of York, where I later then stayed, and I'm currently doing my Masters by Research, where I'm looking at understanding the differences in genetic response to Ralstonia. So we've got tomato, which is susceptible to Ralstonia, and we've got Dulcamara, which is very closely related, which is partially resistant. So Ralstonia is a soil-borne disease that enters the plants via wounds in the roots, and then it travels through the xylem, creates these extra polysaccharides, and then blocks the xylem, blocking the water flow, and then causing these wilt symptoms. So it can infect over 200 different plant species, including tomatoes, potatoes, and eggplants. So we really want to look at different ways that we can really suppress this pathogen. So the experiment that I've been doing, one of the experiments I've been doing has been, someone grew plants for 21 days and then inoculated them with Ralstonia, and then extracted RNA sequence at the time of inoculation, and then again at 6, 12, 24, and 48 hours. Then I took this RNA sequence data and created 2 separate networks, one for each species. So 2 separate weighted gene co-expression network analysis for these 2 different species. And then identified differentially expressed genes at different inoculation status between the 2 species and at different time points to really understand what genes were expressing differently at what times. So this network analysis grouped genes by their expression patterns. So then they'll be grouped into these things called modules, which are also linked to color names. So for example, there'll be a green module, which has a load of genes that are expressed in a similar pattern. So then to really look at those modules that have interesting genes that are potentially expressed differently in the 2 species when inoculated, I had a certain criteria that would have to be met to indicate that they were interesting to look at. So this included DEG movement between our 2 networks. So for example, I've got this graph here at the bottom, which is our Dulcamara network on the bottom, and then our tomato network at the top. And we've got all of our different modules. So if we see, for example, there's a large movement of yellow DEGs into the green module in Dorcomara. So that's an interesting movement of DEGs from one to another, showing that their expression levels has changed to a great extent, that they have changed modules completely. So to quantify this, I also did module preservation analysis, which really gives you some actual values and figures rather than thinking, oh, that just looks like a big shift. Then I also looked at DEG enrichment, so modules that were enriched in DEGs at different time points, especially between the two networks, because that's indicating that there's a temporal shift of those expression levels. I also looked for modules that had interesting gene ontology terms, so terms that were linked to immune and stress responses, indicating that those genes are probably changing due to the inoculation status. So once I found these modules that were interesting, I looked at hub genes, which are genes that are linked to the most amount of genes in that module, because they're hopefully high up in pathways. And then I made sure that they were linked to other GO terms that were linked with immune responses and stress responses. And then I would look closer at their gene expression. So for example, this graph here is split between tomato on the left and Dorcomara on the right. We've got the blue lines, which are infected samples, and the red lines, which are not infected. And this is actually 3 genes, but they all had the exact same expression. So I've put them into one graph. And here, interestingly, we see that there's no expression in tomato, actually meaning that it didn't even turn up in our network analysis for tomato because it didn't fulfill our filtering systems. But then in Dorcomara, interestingly, we see a rise in expression, especially at the 12-hour time point, which does make sense as we're looking at a root-borne disease that eventually makes its way up to the foliar aerial parts of the plant. So we do expect that delay in response in the leaves. And then interestingly, when I used BLAST to identify what these genes were linked to, it was a CDPK1, which is well known to initiate salicylic acid pathways and other immune and stress responses. So that would be a really interesting genes to look further into. So the next step for this one would be looking at modifying this in plantar and seeing if we can upregulate this gene in tomato and see what happens. And also my next step is this, I'm 3 for 3. I've also got a PhD that will be starting in Sheffield in September, which I'm really excited about, on a very similar area to what I've been working on. And the RSB have really helped me with my confidence. It meant that I could start this research as soon as possible, and it's the reason why I've been able to get so much done. I was also able to really improve my lab skills, especially starting my undergrad through COVID. So it's really lovely to be able to have 10 weeks of intense lab work that made me feel a lot more confident, especially doing a lot of DNA sequencing, which I did again for another chapter of my thesis. So that was really, really helpful. And it was really, really nice to work in industry and in a lab and office environment, because it meant that when I started my master's, it felt very comfortable and almost natural to go into that environment. So yeah, I've had a great time, and thank you so much for the RSB and BSPP for funding this position, and I really recommend. I'd also like to thank the Harper Lab, because everyone's been really, really lovely. So thank you.
Thanks so much for the great talk, Kelis. Again, yeah, as you mentioned, great to see all 3 students that have participated going on to further research. Obviously, not everyone will, but it's really great to see how taking part in a research project has helped get students to their next stages in their career and hopefully influence their decisions. So yeah, really great to see some positive experiences. So the last talk for this plant health undergraduate section of today is going to be from a supervisor perspective, actually. So we'll get to see the supervisor side to their story. So unfortunately, they couldn't be with us today, but again, we're keen to participate. So we're going to share the video from Dr. Patricia Ortega-Ramos from Rothamsted Research.
Hi everyone, my name is Patricia Ortega-Ramos and I'm a postdoctoral research scientist at Rothamsted Research. And today I want to talk about cabbage stem flea beetle and how can we tackle them. So first of all, I want to thank the Royal Society of Biology for letting me speak to you today, and also for running the amazing studentships that they run over summer, the plant health undergrad studentships, which I think are a great opportunity for both students and researchers. So I want to take this opportunity to place this project into context and talk a bit about all the activities that we do at Rothamsted and especially at our integrated pest management team. So to start with, I just want to do a brief presentation about Rothamsted. So we are an agricultural research centre based in Harwood, not far from London. We have an incredible experimental farm where we do all our experiments, and we do all sorts of things in agricultural science and innovation. So from soil health and soil microbiology to grazing livestock systems, but also plant sciences and sustainable crop protection and biodiversity, which is what my group is focused on. So our next generation IPM group aims to understand the behavior and ecology of agricultural pests and their natural enemies, and how to apply this knowledge to develop integrated pest management tools to help reduce insecticide use and increase sustainable farming. So our group is focused on oilseed rape, which is one of the most important crops worldwide. It's the third main source of oil in the world. And let's remind ourselves of the main insect threats that oilseed rape faces. So there are a lot of different pests that attack this crop at different stages of the year. So it starts in autumn with the cabbage stem flea beetle, and then it moves on into different species of aphids, then into pollen beetle and rape stem weevils, and later in the spring you can have Brassica podmidge, among others. So it's quite amazing the compensatory ability of this crop and how all these pests try to feed on it and destroy it. And at the end of the season, we still have some crop standing. So what are the control options for all these pests? So in the past, we were using neonicotinoid seed treatments. Unfortunately, these were banned back in 2013 due to the detrimental effects on honeybees and other pollinators. And we were left just with pyrethroids. So these pyrethroids are used in sprays whenever they are needed, and they are the only alternative. However, because of their intense use and the lack of other alternatives, most of the pests I've introduced have developed some sort of resistance. For example, cabbage stem flea beetle resistance was confirmed back in 2018. So, sorry, in 2008. So we've been still using pyrethroids since then, just making the problem of resistance even worse. So this had some effects on the winter oilseed rape, so on the oilseed rape area harvested. You can see in the screen that since 2013 there's been a massive drop in the area harvested both in Europe and the UK, and this has led to a 46% decrease in the area of oilseed rape in the UK and 12% decrease in the whole Europe. It looks like in the last few years there is an increase in the rape area harvested. However, the numbers are still really low and are almost at the same as the ones we had back in 2002. Therefore, it is clear that we need another strategy to manage our crops. We need integrated pest management. And what is integrated pest management? So this is not a single tactic, it's more like a toolbox, many different things that you can do in your crop to manage the pest without going straight into pesticides. So we can describe it with the pyramid that you see in the screen. So we start with prevention, so creating a field environment that reduces pest population and increases natural enemies, or using cultural methods like tolerant varieties, reduced tillage, that can reduce insect pests. Then next comes the monitoring. So if you have a pest problem, you should check the number of pests in your field and compare it with the economic thresholds, and if these economic thresholds are reached, then you can go for interventions. And here we have biotic inputs, so biocontrol agents, and any biological insecticides or even entomopathogenic fungi. And then if there is no other alternative, the last resort will be selective insecticides. And in our group, we are touching almost every single step in this pyramid. So some of the ongoing projects that we have at the moment include habitat diversification effects. So looking into intercropping, undersowing, and flower margins, and how these affect pest and beneficial populations and activity. We also study the insect ecology and biodiversity. So we study their phenology, their life cycle, migration, crop colonization, even their visual ecology and how they can discern between the crop that they want to colonize or others. We also work on push-pull strategies, so identifying plants or cues that can either repel from the cash crop or attract them into other areas. We also work on conservation biocontrol, so it's basically increasing the presence of naturally occurring enemies in our fields. How can we make our fields a better place for them? And also in chemical control, well, synthetic chemical control, so using natural synergies to reduce the use of pesticide, but still controlling the pest. So the work I want to talk about today is this Breeding for Resistance project that we started a few years ago with John Innes Center, with Rachel Wells and Ryan Brook. And this is the main project that Abby Buckle, the student that came with the plant health undergraduate studentships, work on. So the aim of this project was to assess the cabbage and flea beetle damage in 20 different commercial varieties and see if there was any differences. So the final aim was to find this difference and exploiting the genetic variations between them. So we could then go to the breeders and say we found that this variety is performing better against cabbage and flea beetle, and it may be because of these genetic characteristics. So what did Abby did when she wasn't here? So she started assessing the adult feeding damage. So we went out to the field, collected the cotyledons, and took some images of them. And then we analyzed those with image J, so basically counting the number of holes and what percentage of leaf area was eaten. And she found some significant variations between oil-rape cultivars, which is great and is promising for future development. She also looked at the larval antibiosis. So this involved going to the field, collecting plants every month, putting these plants to desiccate, so the larvae can naturally go out of the plant and into those trays that you can see there. And then taking larvae and going through them through the microscope, counting the number of larvae and dividing them between their different instars. So she found significant differences between oil-rape lines between the months. However, these differences were not consistent through the season. However, when looking at the final grain yield, we found that there are some oil-rape varieties that perform better. So considering that we have the same amount of larvae across all the varieties on that season, finding a couple of varieties that perform better means that these varieties are able to tolerate or compensate better for the same amount of larvae, which is really promising. Because she separated the number of first, second, and third instars, she was able to do these graphs and look at how the proportion of first, second, and third instars change across months. So you can see that November will have a similar proportion of first, second, and third, which is expected. But then as we go across the season, so if you go to January, mainly second and third, but the surprise comes in February when we see that the proportion of first instars grows back again. So we think that this can be like the second wave of first instar can be explained because the Cabbage beetle will probably have another egg laying period at some point in February and March, depending on temperatures. And then as you go on March and April, there are mainly third instars, which makes sense because these third instars, as the life cycle shows, then will pupate into the soil. So there, where the asterisk is in the life cycle, there's probably another egg laying period at the end of winter. So as you also looked at the relationship between the number of larvae per plant and the mean larval weight, and she found that whenever you have more number of larvae per plant, these larvae tend to be smaller than when the number of larvae is less per plant. So this is a significant negative relationship between the mean third instar larval weight and the number of larvae per plant. Therefore, it seems that higher larval densities can increase intraspecific competition within the plant, resulting in maybe smaller or less fit adults. So this is something that we want to look into further. And then talking about these follow-up projects, like this relationship between number of larvae per plant and larval weight, we also continued Abby's project with some of the pictures that she took while she was during her time at Rothamsted, to develop some image processing and machine learning tools for automatic identification and quantification of cabbage beetle. So as you've seen during my talk, counting the number of larvae and even assessing the adult cabbage beetle damage is quite labor intensive. So it takes a long time to collect all the plants and then to count all the larvae and identify their instars. So we are trying to make this process much faster and accurate. So with the data that she collected, we are going to hire a new PhD student that will resume this project later in the year. And just to finish with, I wanted to say, well, thank you for the Royal Society of Biology, and thank you for running these studentships, because it's great to have students in research institutes. I think it's a win-win for everyone. Students build their skills, work in a real professional setting, and learn what we do at Rothamsted and how science is done. But also the hosting teams gain some help. Extra pair of hands is always needed, especially during fieldwork, fresh ideas, and a great chance to grow on leadership and project management. And also, it's great to know different people, and Abby was great during her time at Rothamsted. So that's it for me. Thank you very much for your attention, and I hope you enjoyed and found it interesting. And if you have any questions or want to talk more about any of our projects, please don't hesitate to get in touch. Thank you.
Wonderful. I think that was another really great talk to showcase the plant health undergraduate studentships and see the supervisor side. And as Dr. Ortega-Ramos mentioned, you know, the benefit it can have to both the students and supervisors, as the earlier students have also mentioned their experience and skills developed. So that's really great to see. Big thank you to all of the speakers in this session to showcase their research and their experience. As mentioned, please do get in touch with the speakers with any questions that you may have. Do connect with them online if you're interested, and we'll be sure to pass on any questions that you might have for the speakers that weren't able to attend today specifically. But please do check out the Plant Health Undergraduate Studentship scheme on our website if you're interested for next year, either in hosting a project, sponsoring a project, or participating as a student. And if you have any questions about the scheme, we'd be more than happy to answer them.